Good morning, everyone. I am Dr. D. Akila Devi, Professor and Head Department of Pharmaceutical, School of Pharmaceutical Sciences, VISTAS. Today, I will discuss about biodegradable and non-biodegradable polymers and their applications in novel drug delivery systems. Novel drug delivery systems or NDDS are advanced pharmaceutical systems designed to modify the conventional pharmacokinetic and pharmacodynamic behavior of drugs. The primary objective is to achieve controlled drug release, prolonged therapeutic action, and targeted delivery while minimizing the systemic toxicity and adverse effects. In targeted drug delivery, the formulation is designed to preferentially deliver the active pharmaceutical ingredient to a specific tissue or cellular compartment. In controlled release system, the drug is released at a predetermined rate, thereby maintaining drug concentrations within the therapeutic window for a longer period and potentially reducing dosing frequency. Polymers are particularly valuable in this context because their molecular structure can be modified to regulate drug diffusion, degradation, swelling, and membrane permeability. Polymers constitute the structural basis of many modern controlled release and targeted drug delivery systems. As matrix-forming polymers, they provide a continuous network in which the drug is dispersed or dissolved. Drug release may then occur through diffusion, swelling, dissolution, or polymeric erosion. Polymers can also function as rate-controlling membranes in reservoir systems. In such systems, membrane permeability becomes an important determinant of drug release kinetics. A second important application is encapsulation. Polymeric nanoparticles, microspheres, and microcapsules can protect the drugs from environmental and biological degradation and facilitate the sustained release. Polymers can additionally be functionalized with ligands or their chemical groups for site-specific targeting. Stimuli-responsive polymers represent a more advanced approach. These systems respond to physicochemical or biological stimuli such as pH, temperature, enzymes, or light, resulting in controlled drug release. However, irrespective of the delivery strategy, biocompatibility and safety remains essential criteria for polymer selection. Polymers employed in NDDS can be broadly classified as biodegradable and non-biodegradable polymers. Biodegradable polymers undergo chemical or enzymatic degradation under physiological conditions and ultimately generate products that can be metabolically processed or eliminated. This property is particularly advantageous for implantable drug delivery systems because the polymer can progressively disappear after drug release, potentially eliminating the need for surgical retrieval. Examples for applications include temporary implants, injectable depots, microspheres, nanoparticles, and tissue engineering scaffolds. The mechanical system makes them suitable for reservoir systems, long-acting implants, patches, and osmotic drug delivery systems. Non-biodegradable polymers, they remain intact in the body for longer periods, often acquire removal of after drug depletion. They have the excellent mechanical stability and they are preferred for long-term implants and reservoirs, serviced mainly by diffusion method. Let us now consider the major biodegradable polymers. PLGA, polylactic-glycolic acid, is one of the most extensively investigated polymers in pharmaceutical drug delivery. Its degradation profile can be modulated by altering the lactic acid to glycolic acid composition. PLGA is widely used for microspheres, nanoparticles, injectable depots, and implantable systems. PLA or polylactic acid is comparatively hydrophobic and generally exhibits slower degradation than PGA. It has applications in sutures, implants, and scaffolds. PCL or polycaprolactone exhibits particularly slow degradation, often over months or years ago. This makes it suitable for longer duration implants and three-dimensional scaffolds. Chitosan is naturally derived polysaccharide possessing mucoadhesive and biocompatible characteristics. Its cationic nature also enables interaction with negatively charged biological materials and supporting applications in oral, nasal, wound, and gene drug delivery. PGA or polyglycolic acid is more hydrophilic and undergoes comparatively rapid degradation. It is associated with absorbable structures and short duration drug delivery system. Arginate is another natural polysaccharide that undergoes ionic deletion in the presence of divalent cations and is therefore useful for encapsulation and hydrogel based delivery. Applications of biodegradable polymers (NDDS). They have multiple applications. In microspheres and nanoparticles, polymer degradation and matrix diffusion can be exploited to achieve sustained drug delivery of peptides, proteins, and small molecules. For example, PLGA-based lipid technology has been used for prolonged delivery of therapeutically active compounds such as leuprolide. Biodegradable polymers can also be formulated as implantable rods and wafers, where gradual polymer erosion facilitates prolonged drug release. In oral delivery, polymers such as chitosan and alginate can enhance mucoadhesion and provide protection to labile macromolecules against gastrointestinal degradation. For gene and vaccine delivery, cationic polymers can interact electrostatically with nucleic acid and facilitate cellular uptake. They are also valuable in ocular drug delivery, such as ocular inserts, and they prolong drug residence time. Non-biodegradable polymers. So the important examples include silicone elastomers, EVA, PMMA, ethyl cellulose, polyurethane, and polyethylene-based polymers. EVA, that is ethylene vinyl acetate, is particularly used as a membrane-forming material because its permeability characteristics can be utilized for controlled drug release. Polymethyl methacrylate is rigid, transparent, and relatively inert and has applications in bone cements, ocular lenses, and implants. Ethyl cellulose is a water-insoluble cellulose derivative and as an important pharmaceutical film-forming polymer. It is widely employed in sustained-release coatings and matrix formulation. Polyurethane often tunable mechanical properties including hardness and elasticity and are used in mechanical devices such as catheters and drug eluting systems. Applications of non-biodegradable polymers. Non-biodegradable polymers are particularly useful in long-acting drug delivery. In subcutaneous contraceptive implants, silicone or EVA can serve as a polymeric matrix and membrane controlling the release of active agents such as levonorgestrel. In ocular delivery, the occlusal system represents a sustained release approach in which the EVA membrane controls the release of the pilocarpine. Non-biodegradable polymers were also used in transdermal fractures. In osmotic pump system, polymeric components help maintain structural integrity and water influx and drug release. Another major application is drug eluting agents. These materials are particularly useful for extended duration structural stability and controlled diffusion is required. This slide summarizes the fundamental differences between the two polymer classes. In the terms of weight, the biodegradable polymers undergo degradation or elimination, whereas non-biodegradable polymers remain substantially intact. The release mechanism also differs. Biodegradable systems may combine diffusion with polymer erosion, while non-biodegradable systems predominantly depends upon diffusion through the polymer matrix or membrane. The duration of delivery is another important distinction. Biodegradable systems commonly support delivery over days to months, whereas non-biodegradable systems may maintain drug release over months to years. Environmental perspective. This slide provides an additional perspective on biodegradability. Biodegradable polymers such as PLA, PHA, starch-based polymers, PBS, and cellulose can undergo biological or environmental degradation and ultimately generate simpler degradation products. The significance of biodegradability extends beyond pharmaceutical applications because it can reduce the persistence of polymeric materials in the environment. In contrast, polymers such as polyethylene, polypropylene, PVC, polystyrene, polyethylene terephthalate, and nylon exhibit substantially greater persistence. Biodegradable polymers in pharmaceutical applications. From the pharmaceutical perspective, biodegradable polymers offer a combination of biocompatibility, controlled degradation, versatility, and suitability for sustained and targeted drug delivery. PLG is particularly important because its degradation characteristics can be modified and it can be processed to nanoparticles, microspheres, injectable depots, and implants. TCL provides slower degradation and is therefore useful in long-term drug delivery and tissue engineering applications. Chitosan provides mucoadhesion and other functional properties and can enhance its suitability for oral, nasal, wound, and gene delivery. Alginate provides ionic degradation and therefore is valuable for hydrolysis and encapsulation of drugs, proteins, and cells. PGA and PHP or PHA and PBS further expand the range of available polymeric platforms. In biomedical applications, Biodegradable polymers can provide temporary structural support while undergoing progressive degradation. PLG is widely applicable in drug delivery and tissue engineering because of its controllable degradation and biocompatibility. PLA provides relatively high mechanical strength and is useful for biodegradable sutures, orthopedic implants, scaffolds, and stent-related applications. Chitosan provides biodegradability with mucoadhesive, antimicrobial, and hemostatic characteristics supporting applications in wound management and tissue engineering. Arginate is particularly useful in hydrogel formation, cell encapsulation, drug delivery, and wound healing. Other polymers such as PGA, PHP, PHA, and PBS provide additional options depending on the desired degradation profile and mechanical characteristics. Non-biodegradable polymers in pharmaceutical applications, they provide chemical stability and are extensively used in pharmaceutical packaging and drug delivery. Polyethylene offers moisture resistance, flexibility, chemical inertness, and economic make it suitable for pharmaceutical containers, closures, films, and reservoirs. Polypropylene provides chemical resistance, thermal stability, and good mechanical performance and is useful for making syringes, vials, and laboratory containers and other drug delivery devices. PVC polyvinyl chloride provides chemical resistance and useful barrier characteristics and is used in packaging, IV fluid bags, and medical tubing. Polystyrene is rigid, transparent, and readily processed and is widely used in laboratory and diagnostic applications. PMMA provides transparency, rigidity, strength, and biocompatibility supporting ophthalmic and dental applications. Polycarbonate provides impact strength, heat resistance, transparency, and dimensional stability. Finally, silicone or PDMS offers flexibility, biocompatibility, thermal stability, supporting applications in catheters, tubings, implantable devices, and control release systems. Non-biodegradable polymers in biomedical applications. The ultra-high molecular weight polyethylene exhibits excellent wear resistance, toughness, low friction, and biocompatibility. These properties make it particularly important in orthopedic applications such as joint replacement. Polytetrafluoroethylene is chemically inert and possesses low friction and high thermal stability. It has applications in vascular grafts, sutures, and membranes. Polyurethane provides a combination of strength and flexibility and can be used in catheters, tubing, heart-assisted devices, and prosthetic components. Silicone offers flexibility, biocompatibility, and it is suitable for catheters, wound dressings, and soft tissue implants. PMMA is used in bone cement, ophthalmic lenses, dental prosthetics, and cranial implants. Polycarbonate is useful where high impact strength and dimensional stability are required, including surgical and diagnostic devices. Finally, polyethylene terephthalate provides mechanical strength, chemical inertness, and has application in vascular stents, sutures, tissue reinforcement, and medical textiles. Final conclusion. To conclude, polymers provides a versatile platform for engineering modern pharmaceutical and biomedical drug delivery system. Biodegradable polymers such as PLGA, PCl, chitosan, alginate, and PGA are particularly valuable where controlled degradation, sustained release, or tissue regeneration is required. Non-biodegradable polymers such as silicone, EVA, polyethylene, polypropylene, PMMA, PTFE, polyurethane, and PET are selected where prolonged stability, mechanical strength, and long-term performance is required. From a formulation development perspective, polymer selection should be based on drug physicochemical properties, desired characteristics, route of administration, therapeutic duration, polymer degradation characteristics, and mechanical properties, biocompatibility, and clinical requirements. Therefore, the central principle is biodegradable polymers are designed to provide controlled function followed by degradation, whereas non-biodegradable polymers are designed to maintain structural and functional integrity for a longer period. The appropriate selection and rational design of polymers can therefore improve drug stability, controlled release, targeting, and therapeutic efficacy, patient compliance, and overall performance of novel drug delivery system. Thank you.